Everybody see my screen? Okay.
And I will edit this out.
You ready to go?
Jen, it looks like Francis's mic is still unmuted.
Damn it, Frank. Hang on, unmute him. Is that better?
Yeah, that looks good. Or that sounds good.
Okay. All right. If we're good, I'm gonna get started then.
Go for it.
Cool.
We'll have Jen will introduce you.
Okay.
All right. Well, greetings, everyone. If I'm echoing, I'll mute it in a second, and that will stop. But I'd like to thank Lauren for having a seminar. She is a fifth-year graduate student at Rutgers, and Derek is taking pictures. Why? Stop that, Derek. Okay. And so, with no further ado, I'll have Lauren introduce her topic and get started.
Okay. Hi, I'm Lauren Seyler. Thank you, Jen, for introducing me. Today, I'm going to be talking about my dissertation research on marine archaea. Our lab uses a technique called stable isotope probing to explore how microbes, including the archaea, utilize carbon in the environment. And I'll be going over how that technique works and some of the work that I've been doing using that technique to explore archaeal metabolism and metabolic capability in the natural environment, and in particular in a local salt marsh in New Jersey and in the open ocean in the North Atlantic. So mostly everyone is familiar with the archaea, but just as a quick review, we all know the old tree of life with the 5 kingdoms, and the archaea used to be grouped with the bacteria in kingdom Monera. But thanks to Carl Woese's pioneering work over several decades using 16S ribosomal DNA, we now know that the archaea are about as related to the bacteria as we are. They are their own separate domain. And this is an older picture here, so it really only shows the more extreme archaea that we were originally familiar with. So you have your extreme halophiles, your methanogens, your hypothermophiles, and for a very long time we thought that these archaea, the group right in the middle here, were only extremophilic. They didn't live in moderate environments such as the open ocean, but we now know that that's not true. We've found them out in the open ocean and in soils, in sponges, in Arctic environments. Just about anywhere where you might find bacteria, you'll probably find archaea. And the group that I'm going to focus on today is the Crenarchaeota. So just to kind of give you a little bit of a sense of where we're at, you've got your acidophiles up here at the top in this tree, and then over here we have our thermophiles, methanogens down here in the Euryarchaeota, and right smack in the middle, nestled into the thermophiles, are these Crenarchaeota. And they really separate into 2 main groups that I'm going to talk about today. It's marine group 1, also called the Thaumarchaeota, which includes the ammonia-oxidizing archaea. And then down here, the miscellaneous crenarchaeal group, the MCGs. And the name really is indicative of exactly how much we know about them, which is very, very little. They're miscellaneous because they all group together and we don't really know what they do. So, since we've discovered these archaea in marine environments, and they are very difficult to culture, a huge question surrounding them is what exactly are they doing in the environment? And there's a lot of work showing multiple possibilities for their metabolism. There's evidence that they're heterotrophs, that they utilize organic carbon. There's also a wealth of evidence showing that they're ammonia oxidizers, and several ammonia oxidizing archaea have been successfully cultured in the lab. Because there is this evidence of ammonia oxidation, there's a lot of focus on chemoautotrophy in the literature right now because ammonia oxidation is usually linked to converting inorganic carbon to organic carbon. But there's also a good amount of work in the literature right now that suggests that there may be mixotrophs, that they combine heterotrophy and autotrophy, and that they'll do both or one or the other depending on the conditions. And there was actually a recent paper looking at 2 cultured ammonia oxidizers that shows that they are obligate mixotrophs, that they do need a source of organic carbon at least some of the time. So what we can conclude from this body of research is that the crenarchaeal role in the ecosystem is still very poorly understood. We really don't have a firm grasp on their metabolic capabilities yet, and beyond that, we also don't know what exactly their relationship is with the bacteria. Depending on what their role is in the environment, are they competing with the bacteria? Do they have a symbiotic relationship with the bacteria? Do they ignore each other? It's something that we still really need to get our head around. So one technique that I feel will be very useful in answering this question is stable isotope probing. And for those of you who are unfamiliar with the technique, essentially what you do is you provide a substrate to your environmental sample that has been labeled with heavy non-radioactive carbon, so 13C, or you can also use 15N, so you can also trace nitrogen. Whoever takes up this heavy carbon is going to incorporate it into their DNA and produce DNA that now has this heavy label. That DNA can then be separated from the rest of the DNA in the pool, and you can tease out who is actively taking up this carbon substrate versus who is just present. In our lab, we also add a carrier to the separation gradient so that we can visualize a nice thick band of 13C DNA. This carrier is usually belongs to another domain separate from the one that we're trying to look at. So if I want to know who among the archaea is active, I'll use E. coli carrier, for example, so that when we go to amplify this DNA, it won't amplify, it will just be there to visualize the band. You can also add 12C labeled substrate as a control, and then when you go to separate your bands and you pull that bottom band, nothing in the bottom band should amp because it only has carrier and there was no 13C labeled substrate available to be taken up and incorporated into the environmental DNA. Then once you've amplified these bands, you can sort of visualize who's taking up what and who's dormant using a technique like triflip or DGGE, which I'll get into momentarily. So the first study that we did using this technique, we did in a local salt marsh, exotic cheesequake state park in central New Jersey. And we took samples from the levee area, which indicated in yellow on that little map there, which is basically the part of the tidal creek that is intermittently covered with water. When the tide comes up, it's submerged, and when the tide is low, the area is exposed. And we took sediment samples from this area and added them to bottles with sterile filtered site water. We filled them all the way up to the top, unlike in this picture, so that oxygen was depleted very rapidly in these microcosms. And we added 13C substrate or 12C control and incubated these bottles for 5 days in the dark, turning once per day just to make sure everything was nicely mixed together. And the sediment was then collected. DNA was extracted from the sediment and then purified on a cesium chloride gradient. So we have our total population in the blue band up at the top, and indicated in the red at the bottom is our active population in the 13C band. These bands were then amplified, and then we used triflip to visualize the active OTUs and the present OTUs in the sample. So the way triflip works, for those of you who are not familiar, it stands for Terminal Restriction Fragment Length Polymorphism. And how it works is you extract the DNA from your community, You amplify that DNA using 16S ribosomal primers, and the forward primer is fluorescently labeled. So those amplicons are then digested with a restriction enzyme. We usually use MNL1. And that creates fragments that are fluorescently labeled on the 5' end. These fragments can then be separated by size in a gel or a capillary, and then the fluorescence is measured and it creates a series of peaks, each peak representing a different OTU. The height of the peak is equivalent to the brightness of the band as it separates on a gel, and that's roughly equivalent to the number of fragments of that size. So that gives you a rough idea of how much of that OTU, how many members of that OTU are present in the community. However, just because OTUs are all members of, or are all found under one peak, doesn't necessarily mean they're members of the same species. Sometimes you can have two species that both have the an MNL1 cut site at the same location, and so often you will find, depending on the enzyme you're using, you might find 2 or 3 OTUs when you go to make a clonal library that fall under the same peak. But it gives you kind of a general idea of who pops up when, provided a certain substrate or over a certain time frame. So this is what a triplet profile looks like when it comes out. Up at the top we have our control that was fed a 12C substrate. So the blue represents the total population. That would be our top band in the cesium chloride gradient, our 12C band. The red represents the bottom band, which in this case would have only had carrier because no 13C substrate was added to the bottle. And it's a nice flat line, so this indicates that our controls are nice and clean. There's no amplification of that bottom band. There's no incorporation in that bottom band. The two profiles below, the one in the middle represents a microcosm that was fed 30 micromolar sodium acetate, and the one at the very bottom represents a microcosm that was fed 150 micromolar sodium acetate. So there's a 5-fold difference in substrate between the middle and the bottom. And as you can see here, the middle profile has a lot of peaks popping up at this low concentration. But at the higher concentrations, and this kind of held for all of the substrates that we tried, we didn't really see as many peaks, but we did see a very strong signal from certain key players. And so if we take a look here, this kind of pulls together all of our triplet results for all of the different substrates that we used at different concentrations. As you can see on the left here, we had simple organics such as acetate, glycine, and urea, and then more complex mixtures like lipids and isogro, which is a complex growth medium that's labeled with 13C. And just as you saw on the previous slide, at lower concentrations you do see a lot more peaks, but they tend to be very small. So you have all of these little OTUs kind of popping up given a low concentration of substrate. At higher concentrations of substrate, you see these key players with a very, very strong signal pop up, but there are very few of them, and they tend to dominate the entire microcosm. And these were, oh wait, hold on. So we wanted to know, from seeing all of this activity pop up after feeding all of these different organic carbon substrates, if it was possible that the bacteria were incorporating this 13C substrate, producing 13C labeled CO2, and then the archaea were taking up the 13C labeled CO2 and making their own organics from it. Since there was so much in the literature suggesting that Crenarchaea were primarily autotrophic, we wanted to be sure that what we were seeing was true heterotrophy. So we repeated the experiment, and this time we just used 13C labeled urea and 13C labeled bicarbonate. And what you're looking at is just the gel of amplifying the top bands and the bottom bands from those microcosms. So in this lane here, we have no amendment and nothing amplified in the bottom band, as would be expected. In this lane, we had added 12C urea to the microcosm, and again, no amplification in the bottom band. Controls are still clean. This is a microcosm that was amended with 13C urea, and as you can see, we had a nice, strong signal. So the archaea in that bottle were incorporating 13C urea into their DNA. This lane is amplification from a bottle that was fed 12C bicarbonate. Again, no signal, nice and clean. And here's our 13C bicarbonate bottle, and as you can see, there's no band. So we could not detect any autotrophy or any uptake of 13C labeled bicarbonate in this environment over this period of time. So we're fairly confident that the signal that we're seeing is heterotrophy. It is the uptake of organic carbon. And down at the bottom you can see the triplet profile from that 13C bicarbonate bottle, and again, nice flat line at the bottom, and then your active community or your present community in blue. So we did clone some of these active peaks, and what we found were about an even split. There's 14 unique clones that we were able to isolate, and 7 of them were from marine group 1, or the Thaumarchaea, and 7 were from the miscellaneous crenarchaeal group, specifically group 6, or subgroup 6, and subgroup 8. And you can see down at the bottom, N. maritimus in the marine group 1, that's one of the AOAs that are the ammonia oxidizing archaea that has been isolated and cultured in the lab. The thermophiles are on there just as kind of a reference. We didn't find any thermophiles in the salt marsh. And also on this figure, the dark circles next to some of these OTUs represent isolates from other salt marsh samples from the NCBI database. And the open circles represent isolates from the deep biosphere. So it's very interesting that we found a number of OTUs in the salt marsh that did branch somewhat closely to MCGs and MG1s that have been isolated and found in the deep biosphere. So what I'm about to show you now, do we want to pause for questions first? Let's pause for questions first before I move on.
Yeah, and so does anyone have questions for Lauren? Either in the Hangout or on Twitter, please tweet to microseminar or use the hashtag. So Laura, I think, spoke up?
I'm curious how long your tiers of fragments are that we're using to classify the organisms when you clone them.
I could only hear about half of that because of the echo. I'm sorry.
Okay, the echo could be also from me. She wanted to know how long the fragments are.
Oh, for the clones? Yes. So they're about 500 base pairs long. We use crenarchaeal primers 7 forward and 518 reverse. So the fragment when we clone is about 500 base pairs. And then once we clean it up to make the trees and trim out some of the ambiguously aligned sequences, I think the final sequence was a little over 400 base pairs to make the tree.
All right, why don't we keep going?
Okay, cool. All right, so. Okay. So what I'm about to show you is data that hasn't yet been published. We're in the process of writing it up, and it should be ready for submission very soon. So you saw it here first. How exciting. After I did the work in the salt marsh, I got the opportunity to go on a Clivar cruise, the climate variability and predictability cruises. Their main purpose is to study the effects of global climate change, and it's part of the World Climate Research Program. So I was the lone person sampling biomass on the ship, which was an interesting experience. But all along this transect, you can see here, the red dots represent all of the stations that we stopped and sampled at. And the larger yellow dots are stations where I performed SIP. I also took a number of biomass samples from stations along the transect, and these samples were submitted to the Earth Microbiome Project for Illumina sequencing. So I'm going to show you some of that data, and then we're going to talk about some of the SIP work that I've done. Not all of these stations have been processed yet, but the numbers indicated here in white are the stations that have already been processed. And it's kind of a nice little spread because we have a little cluster of stations in the south of the basin, some in the middle, and then some up at the north, and we can kind of compare geographically what's going on. In past studies looking at archaeal distribution with depth, for example in the Pacific Ocean in this paper by Karner et al., we've seen archaea increase with depth up to a certain point, somewhere in the mesopelagic, so around 500 meters, and then stay at about that abundance throughout the water column as you go all the way down to the bottom. And they make up to 40% of the microbial biomass in the water column. What we saw when we looked at the samples that I took on this cruise was a slightly different trend. So the archaea do in fact jump up and increase to a peak somewhere in the mesopelagic, but then they dropped off again. And there's a few spots where they kind of perk up again, but for the most part, the highest abundance of archaea that we found was in the mesopelagic. And as we've seen in prior work, the euryarchaea tend to dominate at the very surface, and then the crenarchaea tend to dominate at depth. Now, these numbers represent relative abundances based on Illumina sequencing. They're not hard counts of cells. So we just finished up some qPCR work in Jen's lab to try to get at some harder numbers concerning, exactly how many archaea are there and how many bacteria are there, so we can get a better idea of whether they're actually increasing or if the total abundances are just, the relative abundances are just changing compared to each other. So using beta diversity analysis and also ANOSIM, analysis of similarity, we saw that there tended to be clustering based on the depth from which samples were taken. So, and this is just representing all of the prokaryotes in the sample. You see there's these distinct clusters of groups, one representing the epipelagic, another from the mesopelagic, the bathypelagic, all the way down to the benthopelagic. So each cluster here represents a different layer in the water column that these samples were taken from. And there's a pretty strong correlation here. And this correlation remains when we split the sample into bacteria and archaea. We see a very strong depth-related clustering for the bacteria by themselves and for the archaea by themselves. There does seem to be some clustering between the bathypelagic samples and the benthopelagic samples in the archaea. They seem to be very closely related to each other. So that's something that I want to explore a little bit further at some point. And if we use ANOVA analysis of variation, to take a look at who is associated with these changes in depth, or who's most likely associated with a particular layer in the water column, there are certain OTUs that are more likely to be found at the surface versus at depth. The ones indicated on the left with the star that says new are ones that were not found in the Green Genes database. The rest are available in the Green Genes database. And all of the ones shown in this graphic represent at least 1% or greater of the archaeal community. There were others that were a little bit more rare that were also tied to this, this association with depth. And we can look here. If we pull out those particular OTUs, and this includes the ones that were less than 1%, and place them in a tree, there is some clustering associated with the sampling depth. For example, there's a bunch of mesopelagic samples over here in the MG1s in the upper right that all kind of clustered together. But it's not really a hard and fast kind of scenario here. There's some mixing. So an OTU that's very likely to be found in the bathopelagic might cluster very closely to one that's more likely to be found higher in the water column in the mesopelagic. And you'll see there's some OTUs here indicated with stars. Those OTUs were also very closely correlated to oxygen concentration, which is interesting because every time I sampled from the mesopelagic, I sampled approximately at the local O2 minimum. So this clustering based on depth and the clustering based on oxygen may be very closely related to each other. It's not strict. But because that local O2 minimum existed in the mesopelagic, then that clustering based on depth may also have a relationship to clustering based on oxygen concentration. So there was a pretty strong correlation for the archaea with O2, and most of the OTUs associated with that correlation were either Thalmarchaea, such as the one that we already have cultured, and Maritimus, and some members of marine group 2, which is a Euryarchaeota. And this correlation also held for the bacteria as well. So oxygen does seem to be a very strong driver of beta diversity in the ocean, and certainly the correlation with O2 was much stronger than the beta diversity correlation with, say, phosphate or nitrate, which are the nutrients that we usually think about when we think about who's driving what's going on in the water column. It's usually abundance of nitrate or abundance of phosphate. So this is basically a summary of this SIP work that we've done so far. And I've separated into the southern stations, which was 30 and 33, the ones in the middle, which is 45 and 48, and the stations in the north, which is station 60 and station 63. We tested so far uptake of sodium acetate, uptake of urea, and uptake of bicarbonate. Unfortunately, we were a little limited on the amount of bicarbonate that we had available at sea, and so we weren't able to test bicarbonate uptake in the bathypelagic. But we were able to take a look at it in the epipelagic and the mesopelagic. And what we found, interestingly enough, is that there doesn't appear to be any OT autotrophic. Certainly many of them are mixotrophic, and at certain depths, certain locations, they will take up one over the other, inorganic carbon or organic carbon. But at many locations and depths, they would take up both. And any OTU that appeared to be strict in one location would take up bicarbonate at another location. For example, if it only took up urea in one, it would take up bicarbonate at another. There were a handful of OTUs, some of the rarer members, who appear to be strictly heterotrophic, like 195 or 158. These were smaller peaks that didn't always show up in every sample. Most of the very large peaks in our triplet profiles appear to be mixotrophic. They would take up both acetate and urea or bicarbonate indiscriminately. And so far we haven't found any particular trend regarding who takes up what where. It doesn't seem to follow any particular pattern that is readily apparent. But I think that there's still a lot left to learn about metabolic capability in the open ocean. And certainly I think we're going to find that it's this metabolic flexibility, this ability to be mixotrophic, that makes them so successful in the open ocean. So in closing, I'd like to thank my PI, Lee Kirchhoff, and technician Laura McGinnis and postdoc Steve Torto at IMCS at Rutgers. Also Michael McCartney and Donglai Gong, who are co-PIs on the Clivar cruise that I participated in. And of course, Jack Gilbert and his tech, Jared Hampton-Marcel, from the Earth Microbiome Project at Argonne National Laboratory. And finally, I'd like to thank you for listening to my talk today. If you don't already, you can follow me on Twitter at Muskamir. I also have a blog on Tumblr. It's mermaidskey.tumblr.com. And I am currently looking for a postdoc position, so if you have a position available, you'd like to talk, my email is here. And I'm looking forward to your questions. Thank you very much.
All right, thank you, Lauren. And if you guys can see us now, you know why I was avoiding doing headphones. This is my grad student, Kristen. We're now sharing earphones.
Hi, Kristen.
So can anyone, if anyone has questions, please let us know. There was a question that came in through Twitter already. And the question is about the correlations. Were they all positive or were they negative? Because you had just talked about a correlation, but we wanted to know if it was a positive or negative one.
Oh, in terms of correlating to depth in O2?
Yes.
So what I found essentially was that O2 and depth have an effect on which OTUs you see in a particular. Hold up a second. Knock my phone.
Lauren, unshare your screen
Oh, sorry about that.
so we can see you.
Okay. Is that better?
Yes.
Sorry. Okay. So, yeah, so depth and OTU had an effect on which OTUs you would see in a particular sample. So certain OTUs were found in a sample that was taken from a certain depth, and then not found in other samples taken from a different depth, essentially. So I guess
So you mentioned that you saw a correlation with certain O2 abundance and dissolved oxygen concentration. So I guess the point of my question was just did you see that relative abundance going up as DO was going down, or did you see the relative abundance going up as DO was going up?
You know, I did check in terms of whether the total relative abundance of archaea was tied to concentration of O2. There doesn't seem to be an actual correlation in archaeal numbers with OTU. In other words, like you won't find necessarily more archaea at a lower at a lower oxygen concentration or a higher one.
Okay.
It just had an effect on which OTUs you would see.
Okay.
Now we did see that peak of archaea in the mesopelagic, which roughly correlates to the local O2 minimum in the basin.
Yeah.
So, looking at it, visually, it would seem that you get higher numbers of archaea where there's lower oxygen. But once you start including samples from the shelf and the slope, that kind of threw off the correlation, I think. So if I went back and just looked at samples from the basin, there might be a stronger correlation in terms of the numbers of archaea.
Yeah, I mean, that would be consistent with other observations that
Yes.
these guys seem to pop up with decreasing oxygen concentrations.
That's true.
Cool.
All right, anyone? I see a few other unmuted mics. Are you ready for questions?
Sure, I've got a question.
Go for it, Mike Wilkins.
I apologize if I missed this early on, but how long, with the stable isotope incubations that you were carrying out, how long were you doing the incubations for? Are they relatively short time periods, or are they more extended?
For the salt marsh samples, they were 5 days. For open water, it only took 2 days to get signal. But archaea tend to be a little bit slower growing. So in our lab, we've been able to get signal in the bacterial populations in hours. But for archaea, we haven't been able to do anything on that short of a time frame. It usually takes, in the open water, it took 48 hours to get good signal.
Okay.
Mike, were you going to go after cross-feeding?
Yeah, I've seen some of the stuff that Lee and his group have done at Rifle, whereby, yeah, you can use a couple of hours to get bacterial labeling. So I was just intrigued how long it took in the open ocean or the salt marshes to get those signals. So yeah, I mean, that was always a concern at Rifle, was that if you left it for too long, because you could get a signal after a few hours, you left it for too long, it would get complicated. Whereas it sounds like you required that amount of time to get a signal.
We really did. I mean, as far as in the salt marsh goes, we've basically ruled out cross-feeding because we didn't see any uptake of of bicarb. Now, that's not to say that the bacteria aren't producing some other byproduct that the archaea are taking up.
Right.
That's possible. At least in open water, I don't think that the archaea are taking up bicarb produced by the bacteria, because in some instances they weren't taking up bicarb at all. Or the amount of bicarbonate being produced, 13C bicarbonate being produced, versus the amount that's already available in the water at 12C, which is easier to take up, would probably swamp out any signal from 13C bicarbonate. So I don't rule out the possibility, though, that the bacteria are taking up these substrates and producing some sort of byproduct that the archaea are taking up and responding to. I don't know how long they would need for that to happen, though. I mean, I used a 2-day incubation time, and past studies of thaumarchaea have shown that they need 2 days to double. And generally, in order to do SIP, you need at least 2 doubling times to get signal, because you have to completely label the DNA.
Mm-hmm.
So we're looking at potentially like a 24-hour doubling time here. So it's possible that the cross-feeding is occurring, but I'm not convinced that it's necessarily happening, especially in terms of production of 13C labeled bicarbonate.
Okay.
It almost works better that they're not all autotrophic.
Yeah, exactly. None of them are coming up as strictly autotrophic, and there were a couple members that were strictly heterotrophic. So it would suggest that if they're taking up bicarb, they are also taking up these organic substrates.
So along those lines, you undoubtedly saw this publication recently in PNAS about the obligate mixotrophic Thaumarchaea. I was just wondering if you had a chance to check and see if any of your OTUs more closely matched those guys than Nitrosopumulus maritimus.
Not yet, but it's definitely on my radar. I just found out about the paper a few days ago.
Mm-hmm.
So
did everybody.
Yeah. So I haven't done that just yet, but it's on my list of things to do.
Cool.
Anyone else in the Hangout have a question? If not, if you're listening through YouTube, please tweet us. And Lauren, if they're tweeting you, I can't see it. So
let me take a look.
You might want to. Well.
No, I don't think I'm getting any questions.
All right. Well, you might after we finish, so hopefully you'll still respond to folks. But with that, I just let you know that we are working on continuing seminars throughout the fall. But because classes are starting and Cameron and I teach on like absolutely opposite schedules, it's going to be a flexible schedule. And so we will rely on Twitter and the WordPress site to get the word out. But if anyone wants to do a seminar, or like Lizzie is going to get forced to do one, please let us know, and you guys know how to find us. All right, so thank you, Lauren. That was an awesome talk, and I think the world of Crens and Uris and archaea in general will continue to expand. Right.
Thank you very much.
So thank you, everyone. I'm going to stop us from going live. Okay. I can figure out how to stop us from going live. What's going on here?
Jen, you're still broadcasting.
I am. How? Why can't I shut it off?
Jen, stop!
You guys are supposed to watch me all day in my office. All right, I'm just going to hang up then. Okay. Thanks, everyone.